Hey guys, welcome back to my channel. So basically today we're going to be going through the topic of microscopes and magnification. So let's get started. So there are two important keywords that everyone has to know for this topic. It's what magnification is and resolution. So magnification is how much bigger the image is than the actual specimen, and resolution is the ability to distinguish between 2 points in an image. So there are two different types of microscopes. One's a light microscope. So a light microscope works by shining light through a very small hole which goes through the specimen. There are lenses that help enlarge and magnify the specimen so we can see it. The specimen is placed on the microscope slide, and light is shone onto the specimen, then has to go through one of the lenses. The lens is called the objective lenses. The coarse focus moves the stage up and down, and the fine focus is for preciseness of the image. So over here you can see the different parts of the microscope as I've just stated. So some advantages of having a light microscope is that it's cheap and portable. So mostly all schools have light microscopes for their experiments, and some disadvantages is that it has very low magnification and resolution, so we can't clearly see the image formed. So it's not very good for universities and scientific experiments. Second type of microscope is the electron microscope. So they work by firing electrons at the specimen, and these electrons have very narrow wavelengths. A more detailed image is produced, and they can magnify something by approximately 2 million times. The specimen is placed in an airtight vacuum. Since it has been in a vacuum, you can only look at the dead specimen, so we won't be able to see the cells moving around. So some advantages are that it has a high resolution and magnification, and disadvantages are that it can be very expensive and you can only see a dead specimen and it's not portable. So this is the required practical of a plant cell and an animal cell. So you can take some time to pause the video and go through it. Okay, so calculating magnification.